Good evening, Dr. Hildebrand. I'm not sure if you're able to unmic. It's at the lower right-hand side of your screen. And if you click that mic, then you should be able to speak.
There we go. Sorry. Is this Victoria?
Yes, hello, welcome.
Hi, Victoria. It's a pleasure to be here. Sorry, I've never used this app before, so I'm still navigating it.
Yeah, well, that's fine. You've opened the room about 38 minutes, 28 minutes, there's, I don't know, 18 minutes early or whatever.
Yeah. Should I close it? I was gonna, Katerina and I.
No, I don't think once it's opened, I think if we close it, then we'd need to reschedule it. So
Got
that's
it.
okay.
Got it.
Yeah, we'll just leave it here. But during the room, you take care of all of the functionality of the app. So yeah, I'll just let other friends, the team, know that you're here.
Okay.
And yeah, good for you. How's your day going?
My day is going well. Yeah. Adjusting. Just got back from travels yesterday, so adjusting back to life, back to reality. Yeah.
Yeah, back to life. Right? Yeah, great. Were you traveling?
Yeah, I was at a conference in Italy actually. So yeah, so I got back from Italy last night to here in Ottawa. So yeah, it was pretty.
Wow.
Yeah, it's great to see the world opening up again. It was nice to see colleagues and old faces and new faces. So it was really good.
Well, that's lovely to get home and have that be welcome.
Yeah, exactly.
Were you there for work? Were you there for a long time in Italy?
Yeah, I was there for a conference, but it was a pretty extensive one. It was 6 days straight, and then I got there early, and I stayed around for a day. So yeah, so it was a decent amount of time to see a bit of Verona. That's where I was, and on the way home, Venice. So it was pretty great.
Oh my gosh, how beautiful.
Yeah, yeah, Venice is just stunning. It's just, and Verona actually. They're both,
Mm.
yeah, so it was really great.
It looks really magical. That's one part of Italy I've never visited, but
Yeah,
yeah, I've seen other people's photos and heard,
yeah,
and so now it's on my list.
awesome.
Mm.
Should I just mute the room? I guess, yeah, now that I've opened
Yeah,
it, or
well, I was gonna say, yeah, you're welcome to just relax, and then we can pick back up in about 15 minutes, and I'll let friends know that we'll be starting at 6. So, yeah, you can just take it easy, relax, get
excellent,
ready, whatever
awesome,
you'd like. And
sounds
then you'll hear one of us saying, "Hello, Dr. Hildebrand." Okay, good. I'll talk to you soon.
okay. Thanks. Okay, bye.
Sure. Oh goodness. Dr. Hildebrand, are you still there? Can you hear me?
I sure am.
Thank you. Can you please click on my profile pic, and then you'll see a drop-down menu open up, and it will say "Make a moderator."
Followed by okay, it's beside it. Make a moderator.
Yeah, if you can click that, then I will have the magic powers that you
I
have, and I
I
can
just
bring.
I
Okay,
just
good. All right. Well, as you were, relax.
perfect.
Thank you.
Thank you.
Uh, hey, Jamie, I've called you up. We're gonna send you an invite, Jamie.
There we go. Hello, Dr. Hildebrand. It's very nice to meet you. Welcome to the Science Society.
Hey, Jamie. Sorry.
Hi, Jamie. It's good to be here. Sorry, I'm just emailing Katerina because she's the one who invited me, and she opened up a side room. But I thought I was supposed to go in this room, so I'm just letting her know where I'm at.
No
Okay,
problem at
so you
all.
still need a welcome. It's good. Okay,
Hi,
Katarina,
Mike.
you're here. So
Hi.
Hey, I'm so sorry. I
It's
sent a link with the wrong date, and I was trying to figure out also what was wrong.
no problem. And then I thought, oh, she opened up the main room early. I guess we're going in there. And then I went in here, so I opened up the main room. So that's my fault. So
No,
we're all
it's
here and we're all
totally my
connected.
fault.
we're all here and we're all at fault.
Yeah.
Sorry.
So then
I sent
maybe,
the
sorry, go ahead,
yeah.
Katarina.
Oh no, it's fine.
I was just going to ask do you still needed to have a welcome room then, Dr. Hildebrand?
No. It was basically just to figure out the functionality of making sure I could speak and navigate the site. But yeah, I think we're good.
Right, good. And then, as I said, we'll take care of inviting up guests and order of speaking and things like that, so you can just relax and enjoy.
Yeah. Actually, the one question I have just in terms of the format, like, because I'm open to questions throughout, like I kind of gather I talk about my research for 20, 30 minutes and there's questions after, but is there a place for people to put like questions in the chat as we go or ask questions, or is it better if I
Well,
just...
that is entirely up to your wishes. There is, if you see on the lower left where it says there's a 3 with a red little dot on top, that's the room chat. And so people will put questions in there, but traditionally we don't expect the guest speaker to be monitoring the room chat. So we
Okay.
would do that and bring up questions to you. You're of course welcome to, but it's our intention that the guest can focus what you're doing best is giving your talk, and then it's up to you if you would like the Q&A to be following your discussion or during your discussion. It's really depends on what makes you the most comfortable and your what you'd like.
Yeah, I can, if people have like a burning question during, they're welcome to ask. I can't see the chat in the bottom here, so I'm wondering, because I'm on a desktop, so I'm wondering if it's...
Oh, I see. And so in that case, also
Okay.
we, as far as us bringing questions, we will save the questions till the end of
Okay,
your
so maybe that's easier.
discussion.
Maybe that's
That's
easier
most common is to do that.
then. Okay.
Rarely people like that,
Yep.
but yes, it's really totally up to you.
Okay.
And you're welcome to just go, until 6. So either way.
Excellent.
Katarina, so you're having trouble with your app.
Yeah.
Okay.
Can somebody pin the link? Yeah, the app is not working. I updated it.
Yes.
Working well.
Okay.
If somebody could try to pin the link.
Yes, got it. I'll do it.
It's let me know if it's working. If not, I have to restart my app, which is also fine.
Show following.
It's done.
Perfect. My app.
Oh, wait.
work,
I
right?
shouldn't have. I spoke too soon. It's not. Let me try again. It wasn't.
The chat.
And then try. Yeah, I copied it. Let me just try it this way. I'm going to try one more time. I don't see any reason why it shouldn't work.
I would prefer to roll back the update. I updated my app. I can now add members and things like that, but then other things don't work well, so I don't know. I thought it
Yeah,
was being
I think a
dumb.
lot of people are having pinning documents up on there as a problem at the moment. I've been hearing it in multiple rooms at the moment, so I think it's just a bug at the present time.
Oh, did you just pin? Is that what you just put in the room chat?
Yeah.
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 Okay. I
So
won't do it then.
people will be able to have the link and look at the paper. It's not like a huge deal, but yeah, it's nicer if it's pinned on top of the room. So, well, Mike, sorry that that my app is having trouble and I kind of screwed up. I'm not sure if the app actually, but it's fine. Thank you for coming and nice meeting you.
Nice to meet you on this platform, Katerina. It's my pleasure. It's my pleasure to be here.
Yeah, thank you so much for doing this. It's I hope you'll enjoy it. Usually people do unless they, They just want to be nice, but usually.
No, I think it's great to find new ways to communicate, and I love this idea of kind of with a lay audience too. So it's really good. Is the typical audience, are there people with a science background, or just kind of any? Like, what's kind of your typical audience member?
It's
Does it
very
vary a
mixed,
lot?
yeah. So,
Yeah.
Cecilia, for example, she is a researcher in public health.
Okay.
Then we have a couple of neuroscientists and engineers. Then
Okay.
Serena is a chemist, but also AI,
Awesome.
AI, and you know, it's very diverse background. And then you know, some people are science enthusiasts and go through Science Society Academy, like Jamie. He
Awesome.
learned
That's
so
me.
much in the last month. It's really amazing to see.
That is
And I'm an art teacher, and I teach different curriculum and focus on science using art. So we are really of diverse backgrounds.
that is really cool. So sorry
you have
for
I beg
I'm
your
just
pardon, Doctor.
I was just seeing the link. Is that the issue, that the paper's not pinned?
Yep, it's supposed to pin at the top where it's supposed to pin above your head.
It says pin a link. Should I try to
Go
paste
for
that
it.
in and see?
Yeah,
If it works for you,
allowing you to
maybe it
save
works
it.
from
But
it works with the desktop app, not with the phone app
ooh,
anymore.
But is that, can you try? That's the, but yeah, I think
that's a paper.
yeah, it's the paper. It's not the PDF version, but then you can click. People can click on the PDF or however they want to.
Let me.
Because it would be good. Because I'm gonna just refer to, like, let's jump to figure 5 type thing.
I'll send you the PDF link.
I can get it if you'd prefer that. I have it here.
No, it's more. It's more.
More accessible. I just didn't know. Okay, so there. So now I have that.
You're already being the moderator. First room already.
Validate.
We already make you work.
Working overtime here. Let's try that.
The desktop app works. The phone. I know that in the morning it worked because I was in the morning listening to the tech newsroom and it worked for them. Unless Tyler does it also on Club Deck on the I don't know. So this is like we are basically a big experiment. This is still like an ongoing development, Clubhouse.
Yeah, which makes it fun.
Yeah, so it's really good because you can actually give feedback and they react quite quickly. Don't you think, Victoria? To like address problems and then communicate.
Yeah, very much. You mean with respect to the app or with respect to the room?
With the app, like with the Clubhouse team, it's pretty.
Yeah, I've found that usually they do respond quickly.
Yeah, the app is in beta still, so it's still being tested and, modified very quickly. I mean, very frequently, actually, like sometimes 2 to 3 times a month, there are updates out.
So my next question is, Bianca, so you saw the room in the hallway. If you want to give feedback in the chat.
Are we concerned that the room is not visible in the hallway?
Yeah, I mean, when I was hanging out in the wrong room I made from introducing to Mike, there were more people just dropping in. That's why we had that issue before. Hi, Serena.
Hello.
Hi, Serena.
We got started early, huh?
Yeah, Dr. Hildebrand is really looking forward to this room.
That's my fault. I was testing things out. Didn't know I opened the whole room up.
20 minutes early. That's a record.
That's a very academic. That's a very academic thing to do before the Zoom meeting. You log in 20 minutes, make sure you can share your slides.
I get it. Yeah.
I was
Yeah.
just thinking that. I was like, he probably thinks, it's kind of like Zoom where you come in early, but no, Clubhouse, once you start it, it's ready to go.
It's ready to go. Everyone can see it. They're like, who is this guy? He obviously doesn't know what he's doing, which is true.
Although you did take care of the pinned link, Serena, he has done the pinned links for us because none of us could do it. So it's good you
I
got
so
there.
get getting there early to make sure there's no tech problems when everyone
Really
arrives.
thoughtful. Most thoughtful guest so far award, I think.
Yay! Do I get a pin for that or a sticker?
Yes, the team will
Well,
discuss it.
actually,
Just you wait and see.
yeah. So, surprise.
Yeah. Great.
Okay, so
So
what
far,
should we do? What should we send him? What do you think? Oh, sorry.
we had we had a few people that we sent stuff because they were just so amazing and nice, and came back a beer glass and a t-shirt were the options so far.
I was teasing.
Science Society merch that we
The
so
swag.
like it's the only two that are out there from Science Society. It's very special.
Yeah, they're limited editions.
Yeah. One of one.
and personalized, of
Yeah.
course. We put the name on there.
And the achievement.
Yeah, that was pretty cool. Like the t-shirt was actually not bad what we wrote on there, but I forgot now. Because it was a room about time travel and being faster than light. So we made like a sentence out of that, but I forgot. I have to look it up. I'm sorry. Getting old.
Yeah, we'll remember it yesterday.
Well, I'm sure today's theme will give us some
Yep.
ideas.
Hopefully it's not interactive, this discussion. Sorry, it's kind of a joke because it's about pain.
Sorry.
Thank you, that's better.
Sorry, I was a little slow on that. It's a little slow on the uptake.
Or I'm not just not that funny.
No, there's no demos. No demos required.
Screaming.
Asking for a volunteer from the audience.
Yeah, you laugh, but when I teach a lecture on this, I actually do. I bring in clothespins, and I get students to put it on their ear. And the whole idea is at the beginning it hurts a teeny bit, but as it hangs there, it just starts to hurt more and more, right? And it distracts you. So to kind of illustrate what chronic pain, not at all to that same level, but the idea how it can change over time and it can,
Yeah, it's great. And develop empathy too for other
Yeah,
people.
exactly. Yeah.
You're much nicer than I, Doctor, because I've got the kind of wicked sense of humour that I would sit a cattle prod down and I would say, who wants to volunteer? Just to see the look on their faces, just for that.
Yeah. I'll have to try that.
Replays are on, everyone. Just so you know, replays are on.
Oh, and Dr. Hildebrand, what that means is once we're done, anyone who wants to go back and listen to this discussion can have access to it on Clubhouse so they can learn from what you're giving here. So it's really, really, really nice.
Excellent. Thanks for letting me know that. That's great.
Have you been having a good evening so far, Doctor?
I definitely have. Yeah, I just got back from my son's soccer game, and now I'm looking forward to getting into chatting some science. So yeah, it's been a good evening.
Oh,
Did they win?
that's excellent. Sorry, someone just asked something, please. Go ahead.
No, I was just asking if they won.
Oh yeah. Did they win, Doctor?
Oh, sorry. Yeah, they did. Yeah.
Yay!
Yes.
That was great. Yeah, they won. I think it was 7-0. But yeah, they just had a turn. They were pretty wiped because they had a tournament on the weekend. So this was their regular season game. So they were running on fumes by the end. But yeah, it was good.
Hey, for everyone in the room, we're gonna start in a moment. We're just sharing the room and having a short discussion beforehand, but we're gonna start soon. So just letting you know.
Ping your friends, friends.
Yeah, thank you, everyone. I think we can actually start. I'm just seeing that I just shared it on Twitter and Instagram and so on. So, yeah, I think we are ready to go. So, thank you, everyone, for coming to Science Society. We are very thankful for our guest speaker, Dr. Mike Hildebrand, who is presenting today his really important research. And let me give you a little bit of information so you get to know him a little bit. He is an associate professor at Carleton University and an affiliate investigator at Ottawa Hospital Research Institute. And Dr. Mike Hildebrand is a neurogeneticist with an international research experience and collaborations. He did his PhD at the University of Melbourne, and he is the head. He had the Translational Neurogenesis Laboratory in the Epilepsy Research Center at the Melbourne Brain Center, and currently he is supported by different major grants working on epilepsy, speech and language disorders, and
That's a different
that's
Dr. Hildebrand.
oh, what?
That's a
Oh,
different Dr. Hildebrand, Katerina, which is all good.
seriously?
Yeah, and it's funny, there's in the same issue of
Oh
Brain,
my
there was
god, this
another
never
Dr.
happened to
Hildebrand.
me.
Yeah, it's all good. I just figure in case people are confused,
Oh
but
my god, oh no, I'm so sorry.
it's totally
Because
fine.
it was also neuroscience, and then
Yeah, and that issue of Brain happened to have another Dr. Hildebrand. Like, what are the odds? Who's very similar.
oh my god, so let's do this as an interview. Okay,
Yeah,
I'm
it's all
really,
good.
really so sorry.
It is
Usually
totally
this is
fine.
the first time. So, Mike, thank you for coming. So, um, if you would like to tell us a little bit about, usually Victoria does this, but how you chose this career in science, and how your path was to become the scientist you are today. Where did you study? So I think this information is really interesting for students that are still choosing their
Yes.
career.
Yeah.
So thank you.
Yeah, I actually kind of stumbled into at least the research path. I was going to teach high school science, and it shows for students out there, sometimes you have kind of one path in mind, and that door actually closed. I didn't get in to that program, but I didn't come from an educated background, and so I learned about grad school and how you could actually get paid to do research as a graduate student. So I applied for scholarships and got into a program to do my PhD at UBC in Vancouver, studying neurobiology, kind of relating to the brain. And then after that, my career path changed. I considered time in industry and spent a little bit of time doing industrial research for a small pharma company in Vancouver. And that's kind of when I thought, let's take the plunge. And when I say it's really my wife, and at that time one kid. We took the risk of kind of pursuing academic research, and we moved to Toronto. And I did further kind of research relating to pain, kind of similar to what I'm doing now. And after that, I was able to get a position kind of where I get to combine kind of my passions of teaching and research. So it's really come together well. And yeah, it's I guess it's a lesson that as long as you're pursuing what you're passionate about, that you don't know kind of where life will take you. And sometimes those detours, actually, they were what helped me get this position, my time in the industry, as well as my passion for teaching.
That's amazing. It takes a lot of courage, I think, to go, to just let your curiosity lead.
Yeah.
So I think that's really impressive. Was there anything earlier in your life that kind of sparked curiosity? Maybe a teacher, somebody in your family?
Yeah, 100%. My high school biology teacher had a big impact on me, and that's when I kind of first fell in love with biology, I'd say. Before that, I was outside in nature, and my dad was really like things like spawning salmon in BC and just learning kind of in an informal way about the natural world. That definitely sparked kind of that curiosity. But then it was a high school science teacher, and then in terms of research, it was honors thesis, a research project as an undergrad student that I wasn't really considering research, but I didn't want to take a certain course, so I took this research project. So for the totally wrong motivation, but I really fell in love. I never expected to kind of enjoy research to the level that I did, and that's kind of what really sparked that idea.
Wow.
That's really cool. I also found in life that when you feel like you're making a major decision, forget about it. Like those tiny decisions that you think are not important sometimes are the major decisions. I don't know, but
Yes.
it's really interesting. That's amazing. I'm glad you found your passion. Not everyone has
Yeah.
that luxury, so
Yeah. I feel very lucky.
that's wonderful. So yeah, that's always so inspiring to hear the stories of people working in science, I think. So thank you for that. And unless does anyone from the moderators want to ask something else before we start with Mike's presentation? Okay.
That all sounds really good. I just want to make a quick announcement that there has been a little trouble on the app with people blocking other people. Sometimes it happens by accident. So we do have a member of our team that's not able to get into the room, and we would sure appreciate if everybody could just double check that that person blocking isn't you, so that we can have Science Society flowing as it should, and everybody can come into this welcoming and inclusive space and enjoy passion for science. So thank you, everyone, and enjoy your talk, Doctor.
Excellent. Thank you so much, Victoria. Okay, so before we kind of dive into what the paper's about and look at some of the results from the paper, I thought it would be good just to provide a little bit of context for this research. So the research is really about chronic pain. And so I guess the first point that we need to consider is the fact that chronic pain is highly prevalent and debilitating. So approximately 1 in 4 to 1 in 5 individuals in society, whether that's the US or Canada or other countries around the world, experience chronic pain. And besides being associated with many diseases, from cardiovascular disease to diabetes to cancer pain to all these conditions that can have this chronic pain symptoms, chronic pain is actually being understood that it can become a disease in and of itself. So even after those triggering events or disease problems or processes, even after they resolve, you can have this persistent chronic pain that can last for months and years and decades potentially. And so obviously this has large economic impacts, just in terms of healthcare costs, lost worker productivity cost. But I think for all of us, what really comes down to is that impact on a personal level. So I bet if I could have a show of virtual hands here and asked who here either directly suffers from chronic pain or has a close friend or family member that suffers from pain, most of us would probably put up our hands. So it's something that I think impacts most of us. And one of the reasons why chronic pain is so debilitating is because we just lack the proper therapeutic tools. We lack safe and effective treatments for chronic pain. And those treatments can include various things like cognitive behavioral therapy, mindfulness, education, physical therapy, many other types of treatments. But my research is kind of more directed at the underlying biology, and so thinking of pharmaceutical treatments that we can potentially develop for pain, because the current ones we have are just very limited. And I think the current opioid crisis illustrates the potential harmful effects of potentially of some of these treatments. So then, how do we get to better treatments? How do we develop as researchers and as a society? How do we develop better treatments? Well, one of the main problems here is we have this big translational gap between preclinical research, which kind of gives us our foundational understanding of how pain works, and clinical research where we can actually develop new treatments. Clinical research that includes the people that need to be treated. And so if we think of this translational divide, on the one side, we have these preclinical models. If we're thinking of pain, we have preclinical models, which are typically often rodents. Historically, a lot of it's been done in male rodents and often young. So our understanding of how pain works is kind of from young male rodents. I'm overgeneralizing. There is some female as well, but that's kind of where a bulk of the research has been done. But on the other side of that divide, when we look at the actual patient population that we need to be treating, the average, at least pain patient, is female. So many pain conditions such as fibromyalgia, irritable bowel syndrome, many other conditions are more prevalent in females. They're obviously human, and the prevalence of pain also increases with aging. So we see there's this discrepancies here between kind of our preclinical knowledge and the patients to be treated. And so the underlying assumption is the drug targets that we're identifying and the drugs we're developing to treat symptoms of pain in our animal models, we're assuming those same mechanisms are at play on the other side in the humans and in females. And so the problem is, for a lot of cases, that hasn't been tested. And so this is the type of research I'm going to be talking about today. That's the idea, to try to start to test those assumptions and see if these pain mechanisms are the same across sex and across species. Okay, and so one more thing before we kind of jump into a little bit about the research results is what type of mechanism of pain are we looking at? We're looking at the spinal cord. And so the spinal cord is a critical hub for pain processing. It connects where your pain detectors are, whether that's in your skin, whether that's on organs of your body, but you have these peripheral sensory neurons called nociceptors that detect those tissue damaging or painful stimuli and encode them into neuronal signals. Then those peripheral signals send that information into the spinal cord. The spinal cord then sends the information up to the brain, which ultimately encodes the unpleasant sensory and emotional experience that we know of as pain. But when I describe that, you could think, okay, the spinal cord is just the telephone lines. It connects where the pain's actually occurring, like on your fingers. If you had a cut, it connects that with the brain, which ultimately will code for that pain. But the spinal cord isn't just telephone lines. It's really a network of processing and turning up and down the gain on that pain signaling. And an example of that is what happens if you're hammering a nail and you hit your thumb. What do you automatically do? And so maybe some of you are doing it right now. You might shake your thumb, or you might rub it. And why do you do that? Well, actually, that rubbing it activates mechanical inputs that are separate. So the touch sensory inputs that are separate from the pain, and those touch inputs activate inhibitory signals that basically turn down the pain sensation. So that's a good example of how pain can be regulated at the level of the spinal cord in the proper context. Today, and what this paper is about, is how when you lose that proper balance between excitation and inhibition, it can cause out of control spinal pain signaling and chronic pain. Okay, sorry, I'm just seeing something pop up, but I don't think that's a question. If you have any burning questions, you can always relay them on to the moderators, and then they can either ask them to me during the presentation or at the end. But okay, so let's go. If you look at the paper then, and I'm just going to really give you kind of a big picture overview of the main findings, and you can always ask follow-up questions as we go through. But we're going to cheat and go right to the end and go to figure 5. And I'm going to do that because I kind of wanted to explain a little bit about the mechanism, so all these specific terms and targets that we're seeing, a bit of them make sense. So if we look at figure 5 and you look at A and B, so A is just a diagram of the type of signaling pathway that we have in these spinal pain processing neurons. And so what happens in chronic pain states, at least for males, and so this was based on a paper previously in 2019 that we published, what happens is that this pain amplifying compound, BDNF, causes a loss of the brakes on excitability, you can think of. And so what that is a downregulation of this molecule, KCC2, which normally keeps the chloride electrical gradient at a certain level. So when chloride builds up in the cell, you lose proper inhibition, synaptic inhibition. And that also downregulates this other molecule, step 61. So those are the brake part of the mechanism. But it's worse than that. Not only do you lose the brakes on excitation, so you have decreased KCC2 in step 61, that directly connects to an increase in excitatory glutamate receptor activity. So you have an activation of this phosphatase or this enzyme known as FIN. And so now, because you have the loss of the brakes, that causes BDNF to subsequently activate FIN. FIN phosphorylates and increases the activity of this excitatory NMDA receptor that's shown now in point 3, the gluN2B NMDA receptors. So you have loss of brakes or loss of synaptic inhibition, increasing excitation. But that was only in males. We saw that in male rats and in male human models. And so actually the impetus for this second paper was actually we started doing, and I'll talk about it more of the human research that we do, but when we were doing that research, we discovered we were doing the human research, and so some of our effects, that decrease in the brakes, increase in gas kind of mechanisms, they got smaller. And so we were really puzzled of what could be going on there. And so I can remember, you have those certain moments in your life, I'm sure you can all reflect back. And I remember driving home and kind of mulling this over in my head of why were these effects getting smaller? And the light bulb going on of, well, obviously we have males and females for our human donors, where all our work in males at the time had been in rodents. And so we went to the results, the Excel sheet, separated it by sex, and saw these huge effects that were in males and weren't there in females. And so that was the launching point for the study, which now we can jump into. I see a hand raised there. I wonder if I can, is that a specific someone asking a question? Can someone jump in just to let me know? Okay.
Oh no, yeah, don't worry about the hand raises. We will take care of that. Thank you so
Okay.
much.
Yeah,
But
yeah.
yeah.
Thank you. Yeah.
Sorry about that.
No,
Okay, I'm learning the app. Thank you for being patient. Okay, so that was a part of Figure 5, A and B, and you might have saw a kind of spoiler alert there, but that's fine too in terms of what's going to happen as we work through the results. But let's go back to the beginning now, and so let's go to Figure 1. Now that we kind of know the context of around chronic pain, needing new treatments, and trying to see if the mechanisms that those treatments might target, if they're the same across sex and species. So the first question then is investigating the spinal mechanism of pain across sex. So using our rodent preclinical models, and in this case, it's a model of inflammatory pain. So I should say, with our animal research, we try to maximize the scientific value while minimizing the impact on the animal, so as non-invasive as possible. So this is a model of inflammatory pain that involves swelling in the paw. So you inject a substance that causes some swelling in the paw, causes hypersensitivity that we can measure. So you see here in A, you see the y-axis is paw withdrawal threshold. So that's basically the amount of force in a thin little filament when you poke the paw that causes the rat to pull its paw away because it's painful. And so you can see here at baseline, there's a certain level, like let's say around 13. But then the dark green, you can see that's after, in the CFA rats, after you've injected that CFA in the paw, you see there's this hypersensitivity. So now a lot less force is required to induce that painful response. And that persists over the next 3 days of testing, day 1, 2, and 3. So in A, that's in males. In females, you see the same thing. You see there's still pain hypersensitivity in these female rats. And I think that is one of the issues with biomedical research, the traps we've fallen into. And I'm not the first. There's lots of research before this. So I actually took this idea from another recent review. But the idea is if we see the same kind of behavioral phenotype or the same outcome, we can assume the underlying mechanisms that got you there are the same. And so that's not always the case. And obviously I'm saying that because if you're looking to your right, you might realize what's leading for me to say that. Because in this case, so we have these models of inflammatory pain, which could be a model kind of like arthritis. It's less invasive than that, but it's that kind of model of an inflammatory pain model. And so we see for the male rats, if you look at B, figure 1B here, you see those markers of the inhibitory mechanisms or the brake pedal mechanisms, KCC2 and STEP on the top, they go down. Specifically, I should say, these are at spinal pain signaling synapses, specifically at the synapses where those signals are coming in. And so you see here, you lose the brakes, KCC2 and STEP for male rats. If you look below that in green, still in 1B, you see the excitatory elements, the phosphorylated fin, I should say, which is active fin, it goes up, and the phosphorylated and total NMDA receptors, the gluN2B, go up. So that's the increased excitation. So you have decreased inhibition in male rats, increased excitation in figure 1B. In female rats, you don't see that. And so it's just striking. If you can see here, NS means not significant. So that when you do the stats, there's no significant changes in those inhibitory markers, KCC2 and STEP. There's no significant changes in the excitatory markers, FIN and gluN2B, in these female rats. So that's biochemical evidence suggesting the pathway that's driving the pain hypersensitivity. Even though there's hypersensitivity in males and females, at the spinal level, what's causing that hypersensitivity in males seems to be different. There's this mechanism there in males that appears to not be there in females. But to more directly investigate that, because this might be, you could argue it's correlative evidence, well, we wanted to directly look at those excitatory synaptic responses, those NMDA receptor responses that we know are critical for regulating excitability in neurons, including these pain signaling neurons. And so now if we scroll down to figure 2, we used a technique called electrophysiology where we can actually record the synaptic NMDA receptor responses from individual spinal neurons. So it's a really powerful technique where we can actually, I always love seeing this, and my excitement never dims of looking at a screen. First of all, you visualize the spinal cord tissue on a microscope. You can bring in a micropipette and seal onto an individual neuron. And then once you've sealed on, you can see the actual individual synaptic responses in real time as they're occurring. And so using that technique, which actually Sackman and Nair won the Nobel Prize for in the 90s, called this patch clamp electrophysiology recording technique, using that, we can see these synaptic responses in figure 2A. And we see, when you look at baseline between males and females, there's no significant difference. So just in the control conditions, kind of normal, quote unquote, conditions, we see there's equal kind of amplitude and equal characteristics between males and female rats for the excitatory NMDA receptors in these neurons. But if we look at our models of chronic pain, the difference here is in figure 2B, you see there's an increase in excitatory NMDA receptor currents in the male rats in that CFA, that inflammatory pain model. Or if you take a kind of in a dish model of chronic pain and just take the spinal cords out of healthy rats and treat them with that substance I was talking about that amplifies pain, that has previously been identified to drive this mechanism. And that substance is called BDNF. So either the in vivo or in living kind of CFA model or the in a dish BDNF model, they both increase the NMDA receptor responses significantly in male rats in figure 2B. Whereas you don't see that in female rats. So that was so here's that clear evidence where there's no potentiation, no increased NMDA receptor responses in female rats in figure 2C here. No significant change. And then once again, just like we did for the CFA inflammatory pain model, we tested for the BDNF in a dish model in female rats. And so in figure 2D, you see there's no change in those inhibitory markers, KCC2 and STEP, or in the excitatory markers, FIN and 2B in these female rats. So once again, in the in-a-dish model, you're not seeing this difference. So we have multiple lines of evidence suggesting this kind of mechanism that not just our lab, but many labs had kind of characterized as being critical for driving increased pain signaling at the level of the spinal cord doesn't seem to be there in female rodents. So at the beginning, I was setting up that if we want new treatments, we need to not just test if these type of mechanisms and potential drug targets. We don't want to just uncover them and investigate them in rodents. We want to see if the same type of mechanisms are occurring in humans. And so I'm really fortunate to be able to partner with a neurosurgeon here in Ottawa that actually I met through a kind of encounter like this. She gave a talk at Carleton University, and Dr. Eve Tsai is her name, and she was like, could you use human tissue? Because she's as a neurosurgeon and with her, she's a clinical researcher as well. She gets human tissue from organ donors. And I should say this is a regulated process that goes after the organ donation process to save lives and improve lives. There's a separate ethical consent that the donors and their families agree to use tissue for scientific research. And we really take this gift as a really precious and selfless gift and try to maximize what we can with this spinal cord tissue. And the real advantage is most human tissue studies are from autopsy autopsy samples that are many, many hours. If they're, for example, spinal cord, it has to be after death. So the tissue can be quite degraded, and it's not as viable to do experiments on. But in this case, actually, Dr. Sion, even my PhD student, Anne-Marie Dedick, who is the lead author on this study, she's actually directly scrubbed in to the OR. So after the organs are removed, that none of that process is compromised. But after that's all done, our team can come in and directly take out that spinal cord. So that means within 1 to 3 hours of the heart stopping, we're able to get that tissue. And because we're able to get it so quickly, it means the health of the tissue is very good. And so we can do the same type of approaches that I've just talked about. And the reason I kind of wanted to walk you through some of those approaches in terms of the biochemistry and that electrophysiology is we're able to do that on human spinal cord tissue of these organ donors, neurologic determination of death or brain dead organ donors. And so using that approach, you can see here in figure 3, if we move to figure 3, we tested whether that same mechanism is occurring in human spinal cord tissue. And this is kind of the first of its kind of investigations into human spinal neurons, pain processing neurons. And what we found is now in the in-a-dish model of pain, where we take neighboring regions of the spinal cord, we treat some with kind of control saline solution, some with BDNF, that pain amplifier, at least in rodents. What do we see? Well, in figure 3A here, we see there's this decrease in that inhibitory marker. In these experiments, we were doing imaging, looking at KCC2, which is that chloride co-transporter, which when it's lost, you lose the proper inhibition. And you see here in the males in figure A, you lose KCC2 at the membrane, and it starts to get trafficked, and it starts to increase in the intracellular compartments, as you see here. And there's pictures in figure 1C of that occurring. I'm not going to go into figure 3C, I should say. Whereas in figure 3B, for the females, BDNF causes no change in membrane versus intracellular KCC2. So you don't see that internalization of KCC2 in female spinal cord neurons that you do see in the male spinal cord neurons. So once again, there's this sexually dimorphic mechanism in human tissue that's the same as what we saw previously in rodents. And then the biochemistry is the same. So in figure 3E, you see this is from the human tissue. When you treat with BDNF, there's no significant changes in those inhibitory, the brake pedal mechanisms, KCC2. And here we have the order wrong step. I just realized for the first time in this paper, I did the order backwards. It doesn't really matter. KCC2 and step don't go down. And the excitatory markers FIN and GLUN2B don't go up here in the human spinal cord, where previously we had already published. We didn't show it again, but in the past paper we showed in the male humans, those markers do change the same way as they do in rodents. So we see the sex difference in pain that's between males and females, where you see this decreased inhibition driving an increase in excitation in males but not females. We see that same mechanism both in rodents and in humans. And so the last experiments were investigating what could be causing that. And based on past studies in the literature, we suspected it could be potentially female sex hormones, but during late development. A lot of the wiring of the brain, there's kind of changes, and it's called masculinization of the brain that's driven by differences in sex hormones between males and females. And I should say these are the biological mechanisms. There's extra constructs that affect this relating to gender, but especially because we're talking a lot of this is animal models, we're more talking about sex in this consideration. But a lot of that happens, those initial differences in the brain and spinal cord happen really early, like just before birth and just after birth. Where in these spinal circuits, actually, a lot of it happens kind of before and after puberty. And so in this case, we took out the ovaries of the rodents before puberty, and in female rodents, and then let those rats mature. And so when you do that, once again, you see no difference. And so figure 4A shows no difference in the synaptic NMDA receptor responses in overectomized versus wild type or untreated female rats. Just like we saw no differences before in male versus female rats. So you see no difference here in figure 4A. But now, when you remove the overectomies, you remove the potential effects of those sex hormones before puberty, and let those rats mature. Now, if you treat the spinal cords from those female overectomized rats, BDNF causes that male-like effect of now BDNF is significantly, so if figure 4B, BDNF in the OVX rats is significantly increasing the synaptic NMDA receptor responses. And that increase can be blocked by blocking one of those excitatory elements, FIN. So PP2 is just a blocker of FIN. When you block that, then when you block FIN, you block the effects of BDNF. So we see the same male-like mechanism if we remove the female sex hormones. So it implies there's a hormonal effect and the effects on the pain circuitry of hormones is what underlies this difference in pain processing. And so that's kind of the main take home. If we go back to now our summary diagram, let's kind of end where we started here on figure 5. And so what the kind of main conclusion here is even though there's pain hypersensitivity in both male and female rats, the mechanisms that drive that can be different. And so in males, we have this BDNF mediated coupling between the loss of brakes, KCC2 and STEP, the increased gas pedal mechanisms, FIN and GLUN2B. You have that in males on the B panel, but you don't have that in females, both for our rodent models and our human tissue models. And that seems to be mediated by female sex hormones. That's what's shown here in panel D. And so we use multiple models of that, inflammatory pain models, in a dish BDNF models. I'm looking at panel E here. But we have yet to investigate whether there's same differences in models of neuropathic pain, which is another type of chronic pain. And the other big question is, well, what's driving that hypersensitivity in females? So follow-up research, we're looking at the underlying mechanisms that might be causing this same increased spinal excitability in females that drives chronic pain. And that could help us understand those differences, identify potential drug targets that work for one or both sexes. And so back to the big picture goal here, we hope this contributes to fundamental understanding that can lead to the understanding of signaling related to chronic pain, and hopefully one day the better and more effective treatment of chronic pain in humans. So I thank you all for your time and patience as we've walked through this, and I look forward to hearing your questions.
Thank you so much. This was such a wonderful talk. And it's really interesting that electrophysiology responses still make you happy, because electrophysiology, when I did a rotation, was what made me stay in neuroscience. It was the coolest thing. You put something on the bath and then it changes.
Awesome.
Fascinating. So
awesome.
It's an immediate reward, right? If you did
Yeah.
it right, you see
You
it
see
right
it.
away.
You see it on the screen. Yeah, it's the analogy of fishing. You have to have patience because it can be frustrating and can take time. But when you get that reward, when you see that response on the screen, there's nothing better. And it's easier for me to say now because I'm not in the trenches. It's my students doing it, so I can walk up and just see the reward without having to have the patience. So that's the one thing I have to remember. Although I should say, with these human tissue, we do. My students and I record together on those because they're such precious tissue that we try to maximize their chance at success.
That's amazing. I never had the chance to record from human tissue, but that's interesting. Yeah, it's cool. And I wanted to ask you, so do you have for any, are you now working on what the female possible mechanism is now, as a follow-up?
Yes, exactly. Yeah. So we're we're looking at the underlying, and there is some other research groups like in the States and other researchers that there's some kind of candidates for what might be driving the mechanisms. There's one compound called CGRP. So the one we talked about today was BDNF, but CGRP, calcitonin gene related protein. It's linked to increased pain processing and preferentially in females, actually. And there is current, in terms of clinical relevance, there are anti-CGRP new treatments on the market right now for migraine, at least. So it's definitely clinically relevant. And so right now we're investigating at the level of the spinal cord whether CGRP might be driving some of these increased excitability in females versus males. So that's one example.
So, is there a way to then block BDNF locally, or is there also because some people get chronic pain, others don't, and some are really extremely at risk, and others are not. Do you think it's a difference in how responsive the neurons are to BDNF, or is there also a difference in some people in BDNF levels?
Those are great questions. So the first one was kind of blocking BDNF. And so that is the tricky thing with any of these kind of, I should say this is one mechanism too. There's lots of other mechanisms. So it's not like I'm saying all pain is caused by this. This is one mechanism that could be contributing. But blocking BDNF, the problem is BDNF has critical roles for learning and mechanisms of learning and plasticity in the brain. So some of these parts of the mechanism we wouldn't want to target therapeutically. But one example in this mechanism of something we could potentially target is that molecule I was talking about, KCC2. It's a chloride cotransporter. When you lose it, you lose the balance of inhibition. And there's actually currently in clinical or at least pharmaceutical development activators or enhancers of KCC2 that are being tested out. So that's one of the parts of the pathway that might kind of be the best fit for potential new treatments. Then you asked about differences between individuals. Absolutely. There can be slight, like what are called as polymorphisms in any of these genes, or environment can sometimes affect, right? There can be epigenetic changes. And so there's definite individualized levels of signaling from person to person for some of these type of mechanisms. And it's not in this mechanism, but an example of that is one of the sodium channels, so that involves in the action potentials, the electrical signaling within pain signaling neurons. What a change in one individual amino acid in that sodium channel can cause this rare genetic syndrome where patients with that mutation lose all ability to sense pain. And then there's other mutations that increase the channel activity, and those ones there's actually patients that have just kind of this persistent chronic pain. So it shows how even small genetic changes in any of these molecules involved in these pathways can definitely cause differences in pain sensation from person to person. So yeah, great questions.
Yeah, thank you so much for answering those. And one last question: is there anyone, like, did people look into if people that have more tendencies to chronic pain also have higher abilities in synaptic plasticity memory, or is it very different in the peripheral? Like, is the neuron type, is it not in general in neurons from a person
Yeah,
basically affected, or?
that's actually a fabulous question. I'd never thought of it. I don't know if anyone, and I think we're too early to know. There are definitely some differences in these spinal circuits, like the types of NMDA receptors. So I was talking about those excitatory glutamate receptors, NMDA receptors we're looking at here. The types that are at these spinal synapses are very different from the brain. They're actually more like the types of receptors that are in the very young brain, where you have increased excitability. And my theory is that's why you have this kind of propensity to lose control of balance of excitability in these spinal pain signaling circuits. So there are some differences between the two. And so maybe it wouldn't carry over, but then there are some properties like these type of mechanisms that do happen in the brain as well. So some things are the same, some things are different.
Is there maybe also is there a mouse model where you could check, there's the switch of GABA to be excitatory to
Yes.
inhibitory, and that's a lot dependent on the KCC.
Yeah,
Um,
exactly. Yep.
is there a difference maybe in, I don't know, in maybe a mouse model that the switch is delayed, or something like that, or maybe earlier on, and that,
And
or
then, yeah,
maybe it stays more excitable in some neurons, that the switch never goes through
yeah,
the whole way.
that's a really good question and good thoughts of, because if you're changing that KCC2, right, that proper inhibition, or at least in later development, proper inhibition is critical for increased excitability early. Does that affect kind of these kind of mechanisms? And I don't know, but that's something we could look at. So that's a really good thought, that we could look at that type of literature and see what's happening. Keeping in mind that's mostly in the brain, those switches. We don't know as much about the spinal cord, and actually what in separate experiments my lab's doing, it looks like the type of molecular switches and developmental switches you're talking about right now that occur in the brain in the first few weeks of life, at least for rodents. For the spinal circuits, it's much later in life. And even that last figure I showed you about the sex hormones is more puberty. And so it looks like actually developmental changes in the spinal cord occur kind of across adolescence into adulthood. And we're thinking that might also kind of shed light on why kind of the prevalence and propensity for chronic pain increases as we age. And so that's another line of research in our lab, is trying to figure out those mechanisms kind of at a molecular level that change across later development in these circuits.
Interesting. So if there's maybe a stressor since there's a longer maybe timeline of fragility or vulnerability, um, would would it be interesting maybe to stress some mice out and then see if that somehow also affects the KCC2 levels?
Yeah, yeah, yep, yep. That could see the because there definitely is research showing that our experience shape us, as we've been talking about, and there's research showing whether it's stressors or even small, like you think of like prenatal babies, how they have all those interventions, right? Like the heel pricks they need for testing. Well, now they're trying to reduce those numbers because it's been shown in humans and in animal models that those small injuries in early life can increase the propensity and prevalence of increased pain responses in later life. And so that's a good point of thinking of the underlying mechanisms of that, how that could kind of relate to things like this, like KCC2. Yes, absolutely. Those are good
Thank
ideas as
you
well.
so much. I'll give the floor to everyone else. Please flash your mic. I have a lot of more questions, but I don't want to take advantage. So, Brian, Abyss, yeah, go ahead.
Hey, Mike, thank you so much for this wonderful talk. It really touched my heart because I actually am a pain patient, and for the last 4 years, I've been reading studies about wind-up phenomena and the dorsal horn and
Yeah,
this vicious substance P glutamate cycle. And I
yeah,
really have to say I've been very confused. It seems like, at least maybe I was reading older literature, there's not that much, from what I could tell, consensus. Please clarify me if I'm wrong about kind of what type of unlearning processes can occur, like if long-term depression happens. And a lot of the papers seem to think ketamine's kind of this like gold standard, this glutamate blocking. But for myself and the other few people with this very rare kind of corneal neuralgia disease, we
yeah,
all did these high dose ketamine infusions, very very high dose, and we all thought we kind of got better, like everything went away. And then within a few weeks of the last infusion, all of us had our pain completely regress. So we're all taking
yeah,
our triptyline or Lyrica or
yeah,
low dose naltrexone. So I was kind of
yeah,
wondering where you thought we stood on, like, is it central sensitization reversible? Does it depend on the patient population, the condition? I mean, I'm kind of confused parsing the literature. I'm definitely no specialist. I'm just a pain
yeah,
patient trying to learn this stuff.
yeah, yeah. No, I think, like you say, a lot of these mechanisms can be very condition and syndrome specific. I should also say up front, I am not a clinician. I have a PhD and mostly animal based research, so I definitely don't want to kind of give the illusion that in terms of medical advice, treatment advice. But what you're saying, I think, could be the case, that you can reverse some of these things, but they can be persistent, right? So during a treatment, something like ketamine, which acts, but we also need to think ketamine acts on many different targets, especially I think at high doses. NMDA is thought to be one of them, but there's other kind of targets that could be involved as well. So during the treatment, it's just like if you have a local lidocaine nerve block, right? Those kind of things, you're dampening that excitability for a while. But does that just dampen it, or does it truly reverse it? And so I think that was kind of the essence of your question. My answer, I guess, is we don't totally know yet, right? And so especially depending on the specific neurons we're thinking about, that I think in some cases that once that excitability, it can be really kind of, it can persist even after once the treatments are removed, that you can have that increased excitability coming back.
Yeah, thanks so much. Is there anything you think like in the pipeline that looks lucrative, like the next 10, 20 years down the road? I think you said something about the gene calcitron. I don't know how to say it, like you said it. I can't remember calcitonin.
KCC2 I was talking
Yeah,
about.
yeah.
Well, CGRP. No, I was talking about CGRP. Sorry. CGRP, calcitonin gene related peptide. That's more for migraine. So that's a good one for migraine. So there's anti-CGRP medications, kind of new frontline medications that I'm pretty sure on the market are close to it. And I think it's not clear how much those will work for pain. And once again, the other thing I should say is I kind of talked about chronic pain as one umbrella term, whereas in reality, and as you know, and I'm sorry to hear of your suffering, Brian, I should say that up front. It can be very debilitating. But every condition can have separate cause. So this is one mechanism, but from a neuralgia to neuropathic pain to fibromyalgia, right, to all these conditions, the underlying mechanisms could differ, or they contribute in different ways. And so I think that's part of it too, is that we need this broad knowledge, but then we need the specific knowledge too, and the more personalized targeted kind of therapeutics of what's occurring in specific conditions, and then in those conditions across variables like different sex and those other kind of things, to really hone it down to figure out what treatments will be best for each individual.
Yeah, thanks so much.
Yeah, and thank you for tuning in, and well done in terms of just your reading and discovery with this. And I don't know if you found there's some really, at least in Canada here, we have some really good patient kind of advocacy and support groups, and some of that can be helpful too, even just to get connected with like-minded.
Yeah, I'm in Toronto, so I'm in touch with Karen Davies and the Canadian
Okay,
Cancer.
yes, excellent. And then not the chronic pain Canada just came online in the last few weeks, so that's I think it's a good online resource that has different things. So yeah, Karen's fabulous.
I believe, yeah, Avis, go ahead.
Yeah. Hi, Mike. Great presentation. Sorry I came in late to the
No problem.
discussion. I don't know if you actually talked about this, but I'm really interested if it's like you mentioned something about removing the different sex hormone generating organs, and then like testing if the BDNF expression is on par with what you see in the wild type. I'm also interested if you actually isolated FSH, like follicle stimulating hormones, and their effect on BDNF expression as well.
Yeah, we haven't done those kind of individual looking at the different hormones. This was kind of really early, and I should say these experiments were done right before the pandemic. It's an example of where the reality of life can impact science. And then our lab got shut down, and so we kind of concluded these experiments because Anne-Marie, the lead student, she was finishing her thesis. So we definitely have follow-up experiments to do, looking at individual hormones like estrogen, FSH, prolactin, and also in males to look at the effects of testosterone, to really try to unpack what's happening to this circuitry across for all of these. But that's a great question.
If I may, is it okay if I, we had a guest speaker here a few months ago. I don't know if anyone else remembers, but he talked about FSH related to Alzheimer's.
Okay.
He was at MROI, and he's now back in his, professor for 20 years, but now he's back in his hometown in China doing research there. And it contributes largely to Alzheimer's in women. So when he blocked that hormone, he could basically, if it was early enough, rescue and avoid like further Alzheimer damage
Okay.
animals. So I don't know if it's directly related, because you said there's a huge difference, or there is a difference between the spinal cord or peripheral neurons and the brain neurons, but I don't know if that could be an
Yeah.
indicator
No.
that it kind of blocks BDNF effect, and then maybe it contributes to Alzheimer's, but also protects from pain. Maybe it's childbirth related. I don't know, something like that.
Yeah, thanks. No, that's a good lead in terms of thinking of, because some of these things in the brain too, and FSH, Alzheimer's, actually one of the other candidates in this pathway, step changes in step balance, can also relate to Alzheimer's as well. So it shows when we have kind of these kind of players that can be involved in many physiological processes, and then when they're not kind of regulated at the right level, can lead to various pathological processes. Yeah, no, that's a great point. Thank you.
I had a question. I'm curious if, and I'm not sure how you'd even do the experiments, but has it been looked at in the human case, in terms of transgender patients, and whether that are taking hormone replacement therapy, whether there's a change in mechanism that is induced over the course of treatment?
No, and that's a fabulous question, Serena. I think this is early. That's an area actually, and Anne-Marie, the lead author on this, who's now postdoc, that's an area she's definitely identified as wanting to take this. We need, for donors, because they're organ donors, they're typically, they're small numbers and just kind of probabilities. We haven't had transgender. But in terms of replacement therapy, that's a good question too. I think it would depend when. Because what we think is going on, that is kind of early development through puberty, that kind of sets the stage. So after that, because for example, our donors, some of them were kind of women, aged women, so it would be menopausal, and we don't see, at least we don't have numbers to do full kind of stats on this, but we didn't see big differences between kind of pre and post menopausal women, at least according to age. We would presume. We don't have the full medical. So I would think that it might be conserved, but that's the kind of research we need in the biomedical field. There's so little on transgender potential differences and hormone replacement therapies. And so maybe we can even use some of our models to look at hormone replacement therapies, even in some of these preclinical models, to at least see and investigate how these mechanisms may be the same or different across gender. So yes, thank you for that great question.
Thank you.
Please flash your mics if you have a question. Dr. Shah, I know you didn't get a chance to ask yet. Jamie, Dr. Shah, go ahead.
Hi, so thank you so much, Mike. That was a fascinating work. I mean, actually, a couple of the points came to my mind after just listening to you. So we know that we have a different type of the KCC. I mean, we have a KCC1 and KCC2, and I was just wondering about, GABA receptor, in relation with the KCC, and did you notice any, changes during, your experiment for the dendritic expression with your, in a rats or not?
Yeah. So the other side, the inhibitory side, in terms of GABA receptors, we haven't looked at that. From what I remember, the literature, I don't think there's big changes, at least in these neurons. So it's more the KCC, the loss of the proper gradient for GABA receptors, because the KCC2, the transporters, and KCC1, the other kind of chloride regulator, seems to be unchanged in these models. So it's more specific to KCC2. And from what I remember, GABA receptor expression doesn't change significantly, but I could be wrong on that. But that's at least. But yeah, that's a good reminder for me to go back and look at that. long term, I should say, even though expression doesn't, you can have at the circuit level changes in the balance between. You can even lose inhibitory interneurons, so GABAergic neurons. Yeah, in terms of the synapse, you can also lose GABAergic synapses. So there are, in other models and other studies, some evidence to suggest those kind of rewiring takes place, but probably more for kind of like neuropathic pain rather than something kind of more acute like these inflammatory pain models.
So do you think that in relation with the interneuron, is that possible that the male, for example, gender more susceptible to the, I mean, kind of things like a seizure or such a thing?
So that would be the brain. Like, would the males be more susceptible to seizure? It hasn't been looked at whether these kind of mechanisms, because some of these elements like ACC2 are linked to, for example, things like epilepsy in the brain. But it has, and I was actually speaking to an expert on that, Kai Kela. He's in Finland. He reached out to me about after this paper came out, and I was asking him whether that's been investigated in the brain. And from what he could recall in terms of potential differences, male female, it hasn't been. So I think that some of the next steps in some of this research is looking at kind of this underlying neurophysiology in the brain, and are there some sex differences relating to some of these things? Because I think I was surprised, at least when I was looking at the literature relating to NMD receptors, like over 90% of the research is all male only, or where they don't say the sex of the animals. So I really think we have a lot of catching up to do, and a lot will be the same, like we've seen shown, right? There will be a lot. You'd expect there to be a lot of things that are conserved, but I think there will be some significant differences that could really help us further understand the processing in the brain as well.
For sure. Thank you so much.
Yeah, Abyss, go ahead.
Sorry, Kat. So I'm kind of curious, like, is there a study that actually looked into the different subunits in the central and peripheral nervous system, and how like BDNF and NMDA receptors actually are sort of purposed in different neurons, either in the central nervous system, can be like cortical systems, or or ganglion in the peripheral nervous system. So I'm just wondering if you looked into the differences in the subunits, or somebody else has.
Yeah, we're doing that actually as we speak. I have a master's student, actually several students on that, looking at the different subtypes of NMDA receptors that are expressed. And so in general, in the spinal cord, so because there are different subtypes, and that has therapeutic relevance because one of those subtypes is kind of highly functional and is really slow decaying, so it really can lead to pain amplification. And that's found in the spinal cord, but not in the brain. It's called gluin 2D. So that could relate. And then your kind of second question is in individual types of neurons in these systems. And yes, that's kind of another technology, single cell RNA sequencing that we can do now, where we can look at the specific genes that are expressed in one type of neuron or cell versus another. And so we've actually partnered with a researcher at NIH, Dr. Ariel Levine, who's an expert in that type of single cell sequencing approach. And so we're doing that on human spinal cord right now to look at the different subtypes. In our case, look at many targets, but in our case, the subtypes of NMD receptors and the levels of those receptors in various different types of neurons in the spinal cord, and comparing that then to male and female rodents, which has previously been done for sequencing, address exactly that differences by cell to cell in the spinal cord. And like you say, that same kind of work can be done and is being done in the periphery by other groups, but not by us.
Would you also be interested in looking in methylation levels around genes that express KCC and the NM?
Yeah, in terms of epigenetic, that would be a good kind of step. We haven't kind of gone there. There's so much to do that, and I don't have that big of a team, and that's kind of beyond my expertise, but that would be a great another thing to do. Yep, methylation to look at kind of epigenetic regulation. We're also thinking splice variants of some of these targets. That's another area in terms of the various variants of some of these receptors and how that could relate to their activity as well. So the genetic approaches for that we're thinking of as well. But we haven't looked at methylation. No.
There's a pretty easy, cheap version to do it. You treat the DNA with, I forgot the name, and it switches out the T.
Okay. Yeah.
I have to look it up. It's really easy, and then you just do sequencing again,
Okay.
and then if it's if that region of interest was methylated or not. Like, it's
Oh,
a really old, cheap version that I use at some
no,
point.
sometimes that's the, I think we sometimes get a little too enthralled with the fanciest and the flashiest tools, but sometimes those old kind of tried and true approaches can be powerful. Well, even some of the techniques here, you see in this paper, right? They're not fancy optogenetics or right, but they can do the job and answer some pretty important questions, Katerina.
It's bisulfite, sorry. I like today my brain doesn't work. It's bisulfite, if you want to
Okay.
look it up. It's
Yeah, I will.
really pretty straightforward.
Thanks.
So I guess I had another question. Last year I was reading Cahal's really old book. I think it's called Degeneration and Regeneration of the Nervous System. And even
Yeah.
there, like over 100 years ago, he speaks about how some type of damage in the periphery can cause a reorganization in the spinal cord. And he speaks all about like these neurotrophic factors and chemoattractants, kind of talking about like this neuroregenerative process from the growth cone when you have these kind of peripheral
Yes.
injuries. And I was wondering, in these kind of animal models where you have kind of all these different methods of causing different kind of insults in the periphery
Yeah.
to cause this type of wind up or central spinal cord dorsal horn pain, has anyone done kind of interventions and maybe even like some type of imaging at the same time where in those interventions they tried to use some type of like, I don't know, like I guess some of the tissue engineering guys, I've seen some papers about like silk threads where they try to create like a nerve growth factor gradient to kind of create like some type of chemoattractant bias similar to I guess what the normal neurotrophic factors would do, but to try to really like aid the regenerative process. And they were actually able to maybe like through that intervention stop that kind of central sensitization in the second order dorsal horn neurons from winding up and the whole kind of inflammatory neuroinflammatory process from beginning. Or is that something that like hasn't had much success, like some type of attempts with interventions in these specific animal models?
Yeah. I don't know if it's being tried in the context of pain because the issue is a lot of those neurotrophins like BDNF, they're increasing, right? So I guess like if you're adding them, you'd be potentially amplifying pain. I think the context of where they can work, or at least they could be more kind of related therapeutics, is spinal cord injury, right? Where you have damage, and that's where the regeneration, right, can happen. And that's, I think, some of the, once again, the yin and yang of those is those factors that can increase the regeneration. Often as a side effect, they can cause the rewiring that will increase the pain as well. So in terms of strategies for that, I don't know if it would work. The one kind of strategy that kind of gets at what you're talking about, the structural reorganization and that's occurring in the pain states, and how do you kind of block that or alter that, are inducing potential stem cells that lead to inhibitory neurons. And so, at least in animal models, there's been a little bit of work relating to that. Alan Basbaum, who's kind of a pioneer in the pain field, he has at least some proof of concept studies where they put in stem cells. I think they're cortical stem cells, though, but inhibitory, and they integrated at least to some level within these dorsal horn circuits, and they did decrease pain. Once again, in rodents and in mice, but at least showing, at least in principle, there might be ways to try to help address, like you're saying, that kind of structural reorganization that can occur to these circuits, that can lead to long-term changes. Yeah. Good question.
Yeah, that's really interesting, and you're absolutely right. Like for my corneal neuralgia condition, there's only a few doctors in the world treating it. The main guy is at Harvard, who I see, and his
Yeah.
treatment plan involves kind of autologous serum or kind of platelet-rich plasma because they both kind of have some nerve growth
Okay.
factors and inflammatory factors.
Okay.
And many patients, including myself, do report an increase of pain while we're on it. But it does seem to be that there is some type of regeneration in the periphery, where even though there's already the rewiring in the central pathways, we do still get some relief. It's definitely not what we wish we were getting, but it does help. So I don't know, just what you said seems to match up pretty well
Yeah.
with like what's happening with the patients right now.
Yeah. And I get what you're saying now. I totally get that. Yeah, you're saying there's damage in the injury, the neuralgia is in the periphery. So those neurotrophic factors are addressing that. But then you're saying, so short term that might work, or well, they could work for addressing that, but it could have other effects too in other parts of the pathway. No, that makes sense to me now. It's really interesting.
Do you expect that the glia cells are also involved in this since yes, I mean since they are involved in probably BDNF level regulation. I'm not 100% sure. I don't know much about the periphery, to be honest. That's
Yeah,
how specialized we are now. So,
yeah, yeah.
um, is there any indications that glia are also, I guess, at least in the inflammatory response, but do they contribute somehow to this chronic pain?
100%. Yes, they release, maybe not in this mechanism. At least that looks like it's prevalent in males. The theory is, at least for nerve injury or neuropathic pain, it's microglia that are releasing the BDNF in males, but not as much in females. But there's also lots of research indicating glial release of cytokines and interactions with NMDA. Actually, the interaction with the NMDA receptors isn't as clear. So that's another direction that our lab needs to still kind of move in, is seeing the glia to neuron crosstalk and how that could be involved. Because yes, there is evidence to suggest they have a vital role and they interact with certain subtypes of neurons within the dorsal horn pain processing circuit. So lots of work to be done there as well. And once again, lots of work, especially when we think of both sexes, because I think a lot of that original work was once again done, probably historically, a lot of it was done in males. So
Very interesting. Thank you so much. I wanted to check with you since we are now going over an hour and it's getting kind of late. If you know, I want to give you the opportunity to get rid of us.
yeah, I have a couple more minutes if there's a couple more questions, and then a bit. Jet lag is starting to kick in. I realized
Yeah.
after, I was in Italy, so the time
Oh
changed.
my
But
God, it's really late.
yeah, yeah.
It's
But
morning.
but this has been really great. This has been a lot of fun. So if there's a couple more questions, I have time to hang around here. But if
Right.
yeah,
Shane, you didn't get that chance, or Wisdom? Would you like to ask a question? Or Jamie, Victoria, Cecilia, him? All are very shy today.
no, I get it.
Shane, yeah,
It's
go ahead.
Oh, I was just going to say, I don't have a question, but this has been a wonderful talk, very informative. Yeah, thank you.
awesome. Thanks for the kind words, Shane.
Yeah, Denise, go ahead.
Same from me. No questions. It was really fascinating. A lot to chew on from my end because this is I'm still learning this stuff. So thank you so much.
Thank you.
Okay. Yeah, then thank you so much, Mike, and I apologize for all the beginning hiccups. I hope you 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 enjoyed it nevertheless, and we really appreciate that you took the time. It was such a informative talk, and it's so important work because I have a dear friend of mine that's also suffering from severe chronic pain, and you know, I heard also from other people in the back channel that listening to researchers like you working on this gives people hope. And also your very inspiring career path that you followed. Your curiosity was really amazing. So thank you so much, and we wish you all the funding and
Yes.
a lot of lab members, and yeah, that everything goes well, and your lab grows and your research grows, and you come back and share updates with us. That would
I
be the
yeah.
coolest thing.
I'd be honored. Thank you. This was a lot of fun. This is my first time on Clubhouse here, and it was a lot of fun. And thank you all for coming out in this evening session.
May I also just say, Doctor, thank you so much for an incredible talk. And I have made a point to contact multiple friends of mine who are suffering from chronic pain, and I'm going to have them listen to this room. And I know they're going to find it very validating and important to know that someone is in their corner and doing this much study and work to helping them. So thank you very, very much for that, and thank you for being here today.
Thank you, Jamie.
Yeah, thank you so much. And thank you, Katarina, for hosting this room. I think because this is so personal for me, this might be the best room I've ever attended on Clubhouse. So really, thank you.
Oh,
Oh, thank you. Yeah, we really appreciate that you came and asked your questions, and they were so interesting. So thank you for being part of this here today.
excellent.
Okay.
Okay. Yeah.
Then have a good night. I hope you don't have too much of a jet lag in the next few days. And yeah, thank you so much. And thank you everyone for coming, asking great questions. Follow the club if you like discussions like this. Maybe we can get Mike back. And, yeah, come back. And yeah, we really appreciate everyone here. And of course, a special thanks to you, Mike. And have a good night, good morning, evening, wherever you are around the world. And yeah, enjoy. Bye.
Okay. Thank you. Bye.
Also, on behalf of Doctor, on behalf of Science Society, please congratulate your son on an excellent win at soccer.
All right. Yeah,
I will do that. He's asleep now, but I'll tell him in the morning that he's
That'll
got
give him
people
something
around
to
the
look forward
world
to.
celebrating that big win.
Yeah,
Absolutely. Thank you.
perfect.
Yes. Take care.
Okay, 3, 2, 1. Bye, everyone.